In this unit, we are going to discuss the various aspects of cellular metabolism, namely cellular respiration and photosynthesis. But first, we need to start with the discussion of what chemistry lies behind reactions involved in cellular respiration. Those reactions, often called redox reactions, are combined of reactions of oxidation and reduction. These two types of reactions are coupled, which means one cannot happen without another. Oxidation reactions are characterized by the loss of an electron from a compound. Here, in the example on the right, A is oxidized, and the reaction that proceeds from left to right is a typical redox reaction. Now, an atom gains an electron in the reduction reaction. So for the B, it is a reduction. It is reduced by A. As we mentioned before, they always occur together. So when the electron is lost, it results in the decrease of potential energy of the compound that is oxidized. In other words, for A, the potential energy is reduced. B, that gains electron, has its potential energy increased. In an example on the bottom, you can see that methane, when it is oxidized and becomes methanol, its potential energy gets reduced. So when you go from left to right, the potential energy of these compounds, it goes down. Essentially, all the energy transfers in the cells occur in increments, and this approach to energy transfer can be only mediated by the transfer of high energy electrons, which means that the energy transfer reactions in the cell are the reactions of oxidation and reduction. Now here you can see that A is oxidized by B, while B is reduced by A. It means that A reduces B, while B oxidizes A, and A plays a role of a reductant, and B plays a role of an oxidant. A, in this case, lowers its Gibbs free energy, and B increases its Gibbs free energy. Now, electrons in the cytoplasm of a cell cannot remain unbounded. So this is why cells use electron shuttles or electron carriers. Examples of electron carriers are provided in the boxes below. First electron carrier is shown here. This is NAD, or nicotinamide adenine dinucleotide. When it is reduced, it accepts two electrons and a hydrogen and becomes NADH, where NADH is the reduced form of the compound. Now, electron carriers are like taxis. They don't keep electrons bound to them for a long time. Their work is to carry and deliver high energy electrons from one member of a biochemical pathway to another. So in this reduced form of NAD, called NADH, when it loses an electron, two electrons, in the reverse reaction, reaction of oxidation, it becomes back NAD+. So this is why NAD+ is called an oxidized form. It is formed in the reaction of oxidation, while NADH is called a reduced form. It is formed in the reaction of reduction. Another example of electron carrier is FAD, or flavin adenine dinucleotide. FAD binds 2 electrons and 2 hydrogens, becoming FADH2. Just like with NAD plus NADH pair, FADH2 here is a reduced form, while FAD is oxidized form. This box generally shows the relationships between the oxidants and reductants, and the box right here just demonstrates that, as a matter of fact, the NAD plus and NADH are nothing more than the nucleoside with 2 phosphates which is bound to nicotinamide. The third type of electron carriers found commonly in plants is NADP plus NADPH pair. Just like with NAD plus NADH, NADP plus is an oxidized form, while NADPH is the reduced form of an electron carrier. The ultimate goal of energy transfer in the cells is to generate ATP. Cells can't store energy in its pure form. Energy is associated with increased temperature, which is associated with the excess movement of molecules. This excessive molecular movement may result in cellular damage. So in order to facilitate energy transfer within the cell, cells use ATP, which is the energy currency of the cell. It's important to understand why cells do not retrieve energy directly from carbohydrates or lipids or proteins. If you think about what happens here, you can quickly understand that if cell tries to get the energy directly from, say, glucose or lipids or amino acids, it needs a set of enzymes that can retrieve that energy from all those compounds. The number of enzymes that's needed becomes unreasonably high. Instead, what cell does, in a limited set of biochemical pathways, it breaks down carbohydrates or lipids or amino acids and retrieves energy from these catabolic reactions. Energy that is produced in those catabolic reactions, these are catabolic reactions that we're talking about. Energy that is produced during these catabolic reactions, exergonic, I want to highlight reactions, is used to generate ATP. Energy of ATP is used for a variety of cellular anabolic reactions, such as formation of polysaccharides or protein synthesis. And the cycle never stops, because when ATP releases the energy, it becomes dephosphorylated. It is broken down into ADP and inorganic phosphate. So, ATP molecules are intrinsically unstable because the phosphate groups, shown here, negatively charged phosphate groups, actively repel themselves. The greatest energy is released when this bond is broken. The breakage of this bond also, so it's pretty much the break when gamma phosphate group breaks off, the greatest amount of energy is released. When beta phosphate group breaks off, still a significant amount of energy is released. In most cases, only gamma phosphate group breaks off of the ATP molecule. Another product of that breakdown is the inorganic phosphate, which allows to explain certain features that occur in the cell that actively use the energy of ATP. So this shows you how the process of ATP hydrolysis and associated energy release is facilitated by enzymes. So molecule A, enzyme, and ATP form a transition complex. In that complex, phosphate is broken away from ATP, resulting in the release of energy. That energy release converts molecule A into molecule B. Enzyme comes out unchanged, and the products of ATP breakdown are illustrated here. So ATP hydrolysis, as we just discussed, releases energy. Now, ADP is regenerated here in the blue reaction, okay, by the reattachment of the gamma phosphate group. Where does the energy for that ATP regeneration come from? It comes from catabolic reactions of breaking down glucose or fructose or galactose or lipids. Essentially, ATP provides an important link between the exergonic and the endergonic pathways within the cell. Now, breakdown of ATP may either result in the release of inorganic phosphate and production of ADP molecule, or it can result in the movement of the transfer of the phosphate group here onto the substrate of the enzyme. Importantly, ADP, a relatively low energy molecule, is formed, and it has to be recycled to produce fresh ATP molecules. The production of ATP from ADP and inorganic phosphate is called phosphorylation. Phosphorylation can occur via two routes. One of them is substrate level phosphorylation. In this case, phosphate is removed from some other intermediate, like this one, and transferred onto ADP molecule, producing ATP and D-phosphorylated product. Now, substrate level phosphorylation has much lower contribution to the ATP production in the cell compared to oxidative phosphorylation, the main component of glucose catabolism that occurs in the mitochondria. So ATP is produced in the process of chemiosmosis on the inner membrane of the mitochondria in eukaryotes, or on the plasma membrane of cells that don't have mitochondria, such as prokaryotic cells. So most of the ATP in the cell, derived as the indirect product of glucose breakdown, chemiosmosis generates about 90% of all ATP in the cell, and production of that ATP is through oxidative phosphorylation. We call this oxidative phosphorylation because oxygen is required for this process. Now, the first step in the process of glucose catabolism is glycolysis. All living organisms carry out glycolysis. What does it tell us? It tells us that the process of glycolysis is evolutionary, very old, very ancient. Glycolysis was most likely carried out by the very first primordial cells. This is why every cell on Earth has the capacity to do glycolysis now. So glycolysis is anaerobic. It can occur in the presence or in the absence of oxygen. It takes place in the cellular cytoplasm, whether we're talking about eukaryotes or prokaryotes. When glucose enters the cell, it utilizes, for the most part, secondary active transport, or it can use facilitated diffusion, getting into the cell through the specialized glucose transporters. Glycolysis can be divided into two phases. First phase, shown here on the left, is energy investment phase. In the diagram on the right, energy investment phase corresponds to the left part of the image. During energy investment phase, the energy of two ATP molecules, I'm going to put them here with a minus sign, is used to activate glucose and convert it into two high-energy intermediates, glyceraldehyde 3-phosphate, or G3P. These molecules now, due to the phosphorylation, have very high Gibbs free energy. G3P are further converted into two 3-carbon molecules of pyruvate during energy payoff phase, shown here. If you look at the diagrams, you realize that for each 2 ATPs spent in energy investment phase, energy payoff phase produces 4 molecules of ATP and 2 molecules of NADH, essentially the ones that carry out high energy electrons. Therefore, the net production of glycolysis is 2 pyruvate molecules, 2 molecules of NADH carrying high energy electrons, and 2 ATPs. How is ATP produced during glycolysis? It is produced via the substrate level phosphorylation. One of the glycolysis intermediates, phosphoenol pyruvate, loses its phosphorus, phosphate group here, becoming pyruvate. This phosphate group in turn is transferred to ADP, generating a molecule of ATP. So what happens to pyruvate next? If oxygen is present, pyruvate enters transition step. Now, obvious question, what if it's not? We're going to talk about it later. So right now, the condition for aerobic respiration to proceed is the presence of oxygen. Pyruvate is converted into acetyl-CoA in the transition step. Now, if you count the number of carbons originating in the original glucose molecule in the pyruvate, 1, 2, and 3, here on top, you can see that. Now, during the transition step, 1 carbon is removed from the pyruvate. In other words, pyruvate is decarboxylated, and it becomes an acetyl group connected to coenzyme A, or acetyl-CoA. So we went from 3 to 2 carbons. Same is exemplified on the diagram to the right. This carboxyl group from the pyruvate is removed in the form of carbon dioxide and replaced by coenzyme A. So you can see the coenzyme A with and without acetyl group at the bottom. So this is the coenzyme A before it has an acetyl group attached. You can see that acetyl group actually represents the very, very small portion of the entire acetyl-CoA complex. This process, transition step, as you can see, occurs in the matrix of the mitochondria in eukaryotes. If we talk about prokaryotes, this process occurs in a cytoplasm, but some next steps will be associated with prokaryotic plasma membrane. This brings us into the conversation about the citric acid cycle, or Krebs cycle. Citric acid cycle occurs in mitochondrial matrix, and it, as any proper cycle, is a loop. The condition for the cycle to occur is the presence of oxygen. However, Krebs cycle does not directly consume oxygen. It generates high-energy electrons, which are carried by NADH and FADH2 and ultimately get transferred to the oxygen. So Krebs cycle overall produces per one molecule of pyruvate 2 molecules of carbon dioxide, 1 molecule of ATP or GTP, 3 molecules of NADH electron carrier, and 1 molecule of FADH2 electron carrier. So here's my highlight. CO2 molecules are shown here. Production of ATP is here. Production of NADH is here, here, and here, and FADH2 is generated right here. Now, I want to sort of consider it head on. You need to appreciate that the production of that the number of Krebs cycles required to process or that occur per one molecule of glucose is 2. If you look at this scheme on the left, you can see that each glucose generates 2 pyruvates. Each pyruvate enters a single transition step. So how many transition steps will happen per 1 glucose molecules? 2. Therefore, how many each acetyl-CoA enters the Krebs cycle? How many Krebs cycles will occur per 1 glucose molecules? 2 cycles per glucose molecule. In the next several slides, we're going to discuss individual steps of the Krebs cycle. I want to highlight you don't need to memorize any formulas of any compounds represented here, but you need to understand the changes to carbons and the products of transition step, glycolysis, and Krebs cycle. Now let's take a look what happens during each step of the Krebs cycle. In the very first step, the condensation step here, the molecule of acetyl-CoA interacts with a molecule of oxaloacetate, seen here. Now, acetyl-CoA has 2 carbons that are active, and oxaloacetate has 4. 2 carbons of acetyl-CoA and 4 carbons of oxaloacetate produce 6-carbon citrate molecule, which undergoes isomerization, producing isocitrate in the step 2. Now, coenzyme A goes back to the transition step and participates in another energy transfer and decarboxylation of pyruvate. Isocitrate in the step 3 is oxidized with the loss of carbon dioxide to produce alpha-ketoglutarate, CO2, and NADH. If you look at the number of carbons, 6 carbons of isocitrate become 5 carbons of alpha-ketoglutarate and 1 carbon in CO2. We talked about the negative feedback regulation of enzymes. Well, it turns out that isocitrate dehydrogenase, enzyme in the step 3, is negatively regulated, inhibited by ATP and NADH, while being positively regulated by ADP. In step 4, 5-carbon alpha-ketoglutarate is oxidized with the loss of CO2 to a 4-carbon molecule of succinyl-CoA. When I use the term 4-carbon, I refer to the carbons that participate in the cycle and not the carbons that belong to coenzymes or cofactors. So during that reaction, another molecule of NADH that carries 2 high-energy electrons is generated. Now, 4-carbon succinyl-CoA is converted to succinate in the process of the 5th step. Okay, substrate level phosphorylation during this step results in the formation of ATP, and then succinate is oxidized. Formation of a double bond is an oxidation. Succinate is oxidized to fumarate. 2 more high-energy electrons are produced, forming a molecule of FADH2. In the last 2 steps, fumarate is hydrated to form malate, and malate is oxidized, that single bond becomes a double bond. Malate is oxidized to oxaloacetate, producing another molecule of NADH. So essentially, we are back to the step 1 of Krebs cycle, step in which oxaloacetate will now react with another acetyl-CoA molecule. When we look at the Krebs cycle holistically, we can now see that 4 carbons that enter the Krebs cycle, 4 carbons that come from 2 acetyl groups in acetyl-CoA, they represent 4 of 6 carbons of a single molecule of glucose. This is why you need 2 turns of the cycle per 1 molecule of glucose. In 2 turns of the cycle, 4 carbon dioxide molecules are produced. By the end of the Krebs cycle, all 6 carbons from the original glucose are oxidized into CO2. 2 of them are oxidized to CO2 during transition step, and 4 of them are oxidized during Krebs cycle. Therefore, in 1 turn of the cycle, in addition to 2 CO2 molecules, 3 molecules of NADH, 1 molecule of FADH2, and 1 molecule of either ATP or GTP is produced. Now, since there are 2 turns required per 1 molecule of glucose, as we discussed, 1 molecule of glucose will generate 6 NADH, this is the typo, 2 FADH2, and 2 molecules of ATP, the products of 2 cycles, and 4 molecules of CO2. Now, Krebs cycle is amphibolic. So what it means is that while the intermediates of the Krebs cycle, such as succinate, fumarate, malate, are essentially the products of catabolic reactions, they also can be at the beginning of anabolic reactions. A lot of intermediates of the Krebs cycle can be used and are used for the synthesis of non-essential amino acids. So far, if you paid attention, you may have noticed that per 1 molecule of glucose, we managed to generate 2 molecules of ATP during the Krebs cycle, and 2 molecules of ATP as a net yield, 2 molecules of ATP here as a net yield during glycolysis, total 4. This is not a lot of ATP for the hassle that we just had to go through. So where do these all what happens? Like, what is the goal of cellular respiration, of Krebs cycle, transition step, and glycolysis? Well, those steps were essential to generate a large number of electrons that will enter the electron transport chain. It is the last step of aerobic respiration. Electron transport chain uses atmospheric oxygen directly, and it essentially consists of a chain of redox reactions. At the very end of these redox reactions is oxygen. Electrons reduce oxygen to water, and the movement of electrons through the electron transport chain is facilitated by 4 different proteins. Electron transport chain proteins themselves are located within the inner mitochondrial membrane of eukaryotes. In prokaryotes, these proteins can be found in the plasma membrane. So there are 4 complexes that form the electron transport chain. We're going to talk about the functions of these complexes one by one. Complex 1 here oxidizes NADH, stripping it of electrons and converting it into its oxidized form NAD+. Electrons enter the electron transport chain at complex 1 and are transferred to the coenzyme Q bound to the complex 2. When electrons move from complex 1 to coenzyme Q, they lose energy. Energy that these electrons lose is used to pump hydrogens across the inner mitochondrial membrane from the matrix into the intermembrane space. In other words, energy of electrons is used to pump hydrogens across the membrane. Now, every 2 electrons will pump 4 hydrogens across the membrane. Now, the function of the complex 2 is to specifically oxidize FADH2 to its oxidized form FAD. The electrons that are stripped from FADH2 enter the electron transport chain and transferred to the cofactor Q, or ubiquinone B. Ubiquinone B, or cofactor Q, or coenzyme Q, connects essentially the complex 1 and the complex 2 to the complex 3. When these electrons, either these or this, eventually enter complex 3 transfers them to the cytochrome C. This transfer lowers the energy of electrons, but this energy is not wasted, because complex 3, just like complex 1, also pumps protons across the mitochondrial membrane. Finally, cytochrome C transfers electrons through the complex 4 onto oxygen, which, when combines with electrons and hydrogens, forms water. Electrons ultimately lose all their energy when they are transferred to oxygen, and that energy that is lost is used with the complex 4 to again pump hydrogens across the mitochondrial membrane, contributing to the gradient of hydrogen ions across the inner membrane of mitochondria. Now that we have discussed the functions of all 4 complexes of the electron transport chain, it's time to ask ourselves a question: what is the purpose of all of this? What is the purpose of pumping hydrogens across the membrane? So, well, turns out that when we pump hydrogens across the membrane, we create what's called an electrochemical gradient. There is an increased concentration of hydrogens in the intermembrane space and the reduced hydrogen concentration within the mitochondrial matrix. So this gradient, so-called electrochemical gradient, across the inner mitochondrial membrane provides the means for hydrogen ions to diffuse through the enzyme called ATP synthase. The diffusion, and you remember from the previous union, the diffusion is the movement of chemicals down its concentration gradient passively. Essentially, these ions, when they diffuse through the ATP synthase, they deliver energy to synthesize ATP from the molecule of ADP and inorganic phosphate. In other words, the potential energy of the hydrogen ion gradient is converted into kinetic energy of hydrogen ions when they diffuse through the ATP synthase. This energy is then used to generate ATP from the molecule of ADP and an inorganic phosphate. So let's take a look at the aerobic respiration. So when you look at the summary, you realize that these are the key products of glycolysis. This is the key product carrying high energy electrons of transition reaction. These are products of Krebs cycle. And this is your total. Now, you understand that by delivering the electrons to the electron transport chain, NADH inevitably provides the energy for ATP synthesis. Turns out that one molecule of NADH provides enough energy to synthesize 3 molecules of ATP, while one molecule of FADH2 provides enough energy to synthesize 2 molecules of ATP. You may ask why. Well, if we go back to this scheme, you can see that NADH delivers electrons to the complex 1, while FADH2 delivers the electrons to the complex 2. It means that the electrons that are delivered by NADH go through more steps in the electron transport chain and release more energy, while electrons delivered by FADH2 use less steps and therefore release less energy. In a sense, you can think of it this way. The electrons delivered by NADH start at the top of the stairs and jump down, roll down the stairs, releasing some energy. Electrons delivered by FADH2, they start not at the top of the stairs, but like at the step 1 or 2. They still roll down the stairs, they still deliver energy, but not as much as NADH does. So when you consider this equation, it's very easy to understand that the total possible theoretical maximum outcome of cellular respiration is 38 molecules of ATP. You can do math yourself. However, it's never 38. Okay? A practical yield of oxidative respiration is always lower than the theoretical one. There are several reasons for that. One, probably the biggest reason, is that we mentioned that hydrogen ions, since they are positively charged ions, can't cross the membrane directly, inner membrane of mitochondria. But it's not entirely true. There are some hydrogen ions that manage to leak out from the inner intermembrane space, they cross the inner mitochondrial membrane and go back into the matrix, bypassing the ATP synthase. In other words, some of the hydrogens that were pumped up here, up in the intermembrane space, using the energy delivered by electrons, some of these hydrogens do not return that energy for the ATP synthesis. They simply sneak through, and that energy is not useful anymore. It is lost as heat, therefore, not all the energy is converted into ATP. And the second pretty important reason why we're not achieving all those 38 ATPs is pretty simple. For instance, pyruvate is to be actively transported into mitochondria. That active transport requires energy. So all in all, the actual outcome, you don't need to memorize that number, but the actual outcome, the actual yield, will be not as much as 38 ATP, but more like 28 to 30 ATP molecules, and in some cases, it is even less. Now, as we discussed the maximum amount of ATP that can be produced during aerobic respiration, we need to ask ourselves another question. Is aerobic respiration the only way to generate ATP, or there are some other processes that can help us in producing ATP? So the answer is there are some others, namely fermentation and anaerobic respiration. So as we just figured out, at the end of the electron transport chain, there are free electrons, and free electrons cannot remain free, so they will be received by the final electron acceptor. So in aerobic respiration, the final electron acceptor is oxygen. In anaerobic respiration, the final electron acceptor is various inorganic molecules other than oxygen, for instance, sulfate or nitrate. I want to specifically emphasize that both aerobic and anaerobic respiration include these steps: glycolysis, Krebs cycle, and the electron transport chain. An example of an electron transport chain in anaerobic respiration is shown in the bottom right corner of this slide. This is the process of anaerobic respiration in the microorganism called Pseudomonas tuceri. You can see that just like in aerobic respiration, complexes of electron transport chain pump protons on the exterior side of the membrane. The big difference, of course, is that instead of cytochrome C oxidase, the transfer of electrons to the final electron acceptor, nitrate, is carried out by a bunch of other enzymes such as nitrate reductase, nitrite reductase, nitric oxide reductase, and dinitrogen oxide reductase. In the end, it is reduced to molecular nitrogen. So, functionally, these processes are essentially the same, but the enzymes within the electron transport chain are different between the aerobic and anaerobic respiration. Interestingly enough, some organisms can perform both aerobic and anaerobic respiration. What if a microorganism or an organism lacks electron transport chain, or for some reason cannot carry it out? For instance, there is not enough oxygen. In this case, some cells can switch to a process of fermentation, where final electron acceptor is an organic molecule. For example, acetaldehyde, final electron acceptor in alcoholic fermentation, or pyruvate, final electron acceptor in lactic fermentation. The reason why fermentation has to be run is to recover NAD+ or FAD from their reduced to the oxidized forms. The image on the left here represents the pathways for ATP metabolism with and without presence of oxygen. So you can see the glycolysis that we've already discussed. And if conditions are aerobic, pyruvate, the product of glycolysis, enters mitochondria, converted to acetyl-CoA, and acetyl-CoA enters the Krebs cycle. All reduced electron carriers are then used in the electron transport chain to produce electrochemical gradient, which drives chemiosmosis through the ATP synthase. Now, in case of anaerobic environment, pyruvate does not enter into mitochondria and instead undergoes fermentation in the cytoplasm. Lactic fermentation, which results in the production of lactic acid, or alcoholic fermentation, which results in the production of ethanol. We are going to discuss these processes in a little bit more details. Fermentation, by definition, is an incomplete oxidation of carbohydrates in the absence of oxygen. The example of lactic acid fermentation is shown on the right. So in the process of glycolysis, glucose is converted into pyruvate, producing 2 molecules of ATP, net yield. During that process, also NADH is produced, but NADH is needed to keep carrying electrons in the cell, so it has to be regenerated to NAD+. It is regenerated by delivering the electrons to the molecule of pyruvate, making it a final electron acceptor in the process of lactic fermentation, and pyruvate by this is reduced to lactate or lactic acid. In the process of alcoholic fermentation, pyruvate first undergoes decarboxylation by the enzyme called pyruvate decarboxylase, producing a molecule of acetaldehyde. Acetaldehyde is reduced, so electrons from NADH go to acetaldehyde. It is reduced by the enzyme called alcohol dehydrogenase and NADH to the molecule of ethanol, and NAD+ is recovered. This illustrates, again, that NADH has to be recovered and that acetaldehyde is the final electron acceptor. As you of course know, alcoholic fermentation is used for the production of various alcoholic drinks, such as wine, beer, and lactic fermentation is widely used for the production of certain foods, for example, sauerkraut. Now, what I want you to appreciate is that the amount of ATP that's produced during fermentation is lower than the amount of ATP produced during respiration. And specifically about respiration, anaerobic respiration produces less ATP than aerobic respiration. Now, so far we were talking only about the metabolism of carbohydrates. Now, what about the metabolism of lipids or proteins? Can they be used as the energy source? The short answer is yes, they can be. And moreover, they will be the products of the metabolism will be, as I call it, plugged in to the metabolism of carbohydrates. In other words, catabolic pathways for carbohydrates, proteins, and lipids are all interconnected and are reversible often. So here in the center, you can see what we just discussed. Monosaccharides in the process of glycolysis are converted into pyruvate, which undergoes the transition step and the resulting acetyl-CoA enters the Krebs cycle. Now, triglycerides are broken down into glycerol and fatty acids in the process of lipolysis. Glycerol can be converted into glyceraldehyde 3-phosphate, and fatty acids are oxidized to acetyl-CoA by the process known as beta oxidation, which occurs in mitochondria. Proteins are broken down into amino acids, which in turn undergo deamination and transamination. You don't have to distinguish this, but deamination and transamination results in the production of either a molecule of pyruvate, which then can be converted to acetyl-CoA, or directly some of the intermediates of the Krebs cycle, such as oxaloacetate. Pathways that can be reversed are often called amphibolic pathways. Now it is time to talk how all those carbohydrates can be produced by the, well, producers in the first place. We are going to talk about photosynthesis. So it is a biological process that converts the energy from the sunlight into the chemical compounds. So again, energy is not created nor destroyed. Also, one of the products of photosynthesis is oxygen, which obviously is necessary for living organisms. How does it work? So the energy of light energizes electrons, which, as we mentioned before, carry energy. The energy of these electrons is stored afterwards in the sugar molecules. The organisms that can carry out photosynthesis include plants, algae, and cyanobacteria, which are called photoautotrophs. So photo refers to light, auto refers to self-feeders. They produce organic compounds from inorganic. On the other hand, animals and fungi and most overwhelming majority of protozoa and other organisms are heterotrophic. In other words, they consume organics produced by autotrophs. They rely on carbohydrates that are generated by the previously mentioned photosynthetic organisms in the process of photosynthesis. Interestingly enough, some bacteria can feed themselves, being autotrophs, but use not the energy of light, but instead the energy that they can derive from other inorganic compounds. Bacteria on the right are found near volcanic vents at the bottom of the ocean, obviously the place where sun never shines, but very rich in terms of high energy inorganic compounds. We call these organisms chemoautotrophs. Photosynthesis itself is a process that consists of multiple individual steps. First of all, it requires specific wavelengths of light. carbon dioxide, and water. The two immediate products of photosynthesis are oxygen and glyceraldehyde phosphate, which eventually can be converted into sugar molecules. If you go back to this scheme, you can see that glyceraldehyde 3-phosphate in the center of the slide. If you run the reaction of breakdown glucose backwards, you can use glyceraldehyde 3-phosphate to synthesize glucose instead. Photosynthesis occurs in the leaves of the plants, in their middle layer, so-called the mesophyll layer. It is perfectly illustrated on the left. You see the tree on the left, and you can acknowledge that sugars are made in the leaves. As we mentioned before, photosynthesis requires water. Water is delivered via the roots to the leaves. Carbon dioxide enters the leaves from the atmosphere, and oxygen that is produced during the process of photosynthesis is released from the leaves back into the atmosphere. Carbon dioxide and water enter, well, carbon dioxide enters while oxygen exits through the openings in the leaves called stomata. Stomata not only regulate the gas exchange, but also regulate water balance. When they open, water can evaporate from the plant, and when they closed, it cannot. Typically, stomata found on the underside of the leaf to better control the gas exchange, water balance, and prevent overheating, prevent the impact of the light on the gas exchange, and prevent excessive water loss. Opening and closing of stomata is controlled by guard cells. Within the mesophyll of a leaf, you can find an organelle that essentially carries out the process of photosynthesis, a chloroplast. It is enveloped by two membranes, the outer and the inner, and contains stacks of membrane organelles called thylakoids. So each stack of thylakoids is called a granum, grana for plural, and grana are surrounded by the gel-like fluid called stroma. In the membrane of thylakoid, you can find a bunch of pigments, chlorophylls. The role of the chlorophyll is to absorb light. You can also find electron transport chains in the membrane of thylakoids. So the reaction for the photosynthesis is shown in the left. Carbon dioxide and water are used to generate sugars and oxygen with the utilization of the sunlight energy. In general, not in general, but always, photosynthetic reactions can be divided into two broad categories, light-dependent and light-independent. And the name shows you that light-dependent reactions rely on the light energy to occur, while the light-independent reactions, also known as the Calvin-Benson cycle, do not. During light-dependent reactions, sunlight energy is converted into chemical energy. This chemical energy, presented here as molecules of ATP and NADPH, and shown on the left as the same molecules, this chemical energy is then used in the Calvin cycle, or Calvin-Benson cycle, to drive the conversion of carbon dioxide into sugars. This process is called carbon fixation. So the role of ATP and NADPH is to serve as electron or energy carriers to deliver electrons and energy from the light-dependent reactions in the thylakoid to the Calvin cycle in the stroma. Now, what is light? Light is an electromagnetic radiation. It travels as waves, and waves can be characterized by the wavelength and the frequency. So wavelength is the distance between two crests or two troughs of a wave, and the shorter the wavelength, the more energy it has. Waves with super short wavelengths, such as X-rays, UV light, and gamma rays, can be so high energy that they can present danger. The scheme below shows you the distribution of wavelengths across the electromagnetic spectrum. This little part of the spectrum is actually visible light. When you look at the image, you quickly realize that violet and blue colors have the shorter wavelengths, meaning higher energy, while orange and red colors have the lowest wavelengths and their longest wavelengths, and therefore lowest energy. Now, what is the role of pigments? Well, pigments found in plants, algae, and cyanobacteria absorb specific wavelengths of the visible light. And as you may remember from the previous slide, those wavelengths lie in the range between 7 and 400 nanometers. If the energy of the photon that is absorbed by a pigment is sufficient, it will raise an electron from its ground state to its excited state. This radiation with enough energy to excite an electron is called photosynthetically active radiation. As we mentioned before, shorter wavelengths correspond to the larger, greater energy of photons, but it doesn't necessarily mean that only shorter wavelengths will be used in a photosynthesis. In photosynthesis, each pigment absorbs only a specific set of wavelengths. And the wavelengths that they cannot absorb, the pigments will reflect or transmit. Two pigments that we are interested in here are chlorophylls, and on the image shown as A and B, and carotenoids. There are 5 major types of chlorophylls: A and B found in plants, C and D found in algae, and bacterial chlorophyll that's found in cyanobacteria, prokaryotic photosynthesizers. Carotenoids are more numerous, and they're mostly found in fruit. For instance, lycopene is found in tomato, zeaxanthin found in corn, and beta-carotene found in oranges. The main function of carotenoids is to dispose the excess light energy as heat, not so much participate in photosynthesis, but protect plants from excessive electromagnetic radiation coming from the sun. Every pigment, whether it's a chlorophyll or carotenoid, can be identified by the specific absorption spectrum. If you take a look at the absorption spectrum of chlorophyll A, shown here as the solid line, or chlorophyll B, shown here as the dashed line, you can see that they absorb mostly in blue and red regions, which explains why chlorophylls are green. This is what gives color to the tree leaves. Carotenoids absorb almost exclusively, carotenoids represented as a dotted line, almost exclusively in the blue and purple regions of the spectrum, which explains, and a little bit in the green, which explains why carotenoids in general have yellow, red, or orange appearance. Now, this mixture of pigments in photosynthetic organisms allows them to use a wide range of wavelengths for the photosynthesis. Moreover, plants that are in the shade can adjust the relative concentrations of different types of chlorophyll to make the light absorption most efficient. So what essentially happens in photosynthesis when light is absorbed? So light-dependent reactions of photosynthesis depend on two photosystems. Photosystem 2 that engages first, and photosystem 1. And these photosystems convert the energy of light into chemical energy of ATP and NADPH. Each photosystem is essentially a multi-protein complex, but the structures of photosystem 2 and photosystem 1 are relatively similar. So-called antenna proteins form so-called light harvesting complex. Within antenna proteins, you can see individual pigment molecules which absorb photons of light. Absorption of a photon excites the electrons in the pigment molecule, and the energy of excited electrons is transferred between the pigments until it reaches the special pair of chlorophyll molecules in what is called a reaction center of the photosystem. The role of that energy that is delivered to the reaction center of the photosystem 2 or photosystem 1 is to energize an electron pair from the reaction center to deliver it to the primary electron acceptor in the corresponding photosystem. So the first photosystem that we're going to talk about is actually a photosystem 2. In this image, photosystem 2 is shown on the left, and the chlorophylls in the reaction center of the photosystem 2 lose the electrons. They are essentially oxidized. When they lose the electrons, the name of that moment is a photoact. The electrons that have been energized in a photosystem 2 transfer from reaction center to plastoquinone and then to the cytochrome complex. The role of the cytochrome complex in a photosystem 2 is to move the electrons further to the molecule of plastocyanine. Every movement of electrons facilitates the pumping of hydrogens across the membrane into the inner space of thylakoids. You've already seen that, haven't you? This is an electron transport chain. So the role of high-energy electrons that are essentially energized using the energy of light in the reaction center of photosystem 2 is to give that energy away in the electron transport chain so that this energy can be used to move hydrogens into the thylakoid space across the membrane, essentially creating electrochemical gradient of hydrogen ions. Now, where do these electrons go after they arrive to plastocyanine? After plastocyanine, these electrons enter photosystem 1. We're going to talk about what happens there, but for now, let's focus on photosystem 2 or P680 photosystem. So when these electrons here, when they go missing, they have to be replaced. These electrons are replaced by oxidizing a water molecule. This oxidation results in the production of hydrogen ions and oxygen. Photosystem 2, meanwhile, is reduced, re-reduced, and ready to absorb the energy of another photon. Out of all oxygen that is generated during the light-dependent reactions of photosynthesis, about 10% is used by the mitochondrial plants for the aerobic respiration. The rest of this oxygen is used by other aerobic organisms, such as heterotrophs. And as we mentioned before, the energy of electrons that go through the electron transport chain is used to produce the electrochemical gradient, essentially high concentrations of hydrogen ions in the thylakoid lumen compared to the stroma. Now, photosystem 1 conceptually performs the same. So photosystem 1 re-energizes the electrons that have lost their energy by absorbing another photon of light. Just like with photosystem 2, energy is relayed to the reaction center by pigment molecules bound to the antenna proteins and light harvesting complex, and photosystem 1 is oxidized again. When it is oxidized, these electrons are sent through NADP+ reductase to the molecule of NADP+. NADP+ electrons and hydrogen ions combine together, forming a reduced form, NADPH. So right now we can summarize the functions of photosystem 2 and photosystem 1. When photosystem 2 captures the energy of light, this energy is ultimately used to generate a proton gradient, which will be used afterwards to make ATP. Photosystem 1 captures the light energy to energize electrons so they can reduce NADP+ to NADPH. The amount of NADPH produced by photosystem 1 and the amount of ATP produced by photosystem 2 are roughly equal. So this picture is familiar to you since we saw it before when we discussed cellular respiration. Elevated hydrogen concentrations within the thylakoid lumen furnish a concentration gradient which is directed from the thylakoid lumen to the stroma. Hydrogen ions diffuse passively through the ATP synthase from thylakoid space into the stroma in the process of chemiosmosis. That diffusion provides energy for the synthesis of ATP by ATP synthase. This process is essentially the same as the process of ATP synthesis in electron transport chain and chemiosmosis of the cellular respiration. Now we harnessed a whole bunch of energy in light-dependent reactions. This energy we just used to produce ATP and NADPH. Now, these two molecules will be used in a Calvin-Benson cycle, which occurs in the stroma of the chloroplast, to convert carbon dioxide ultimately into organic compounds. So how is carbon dioxide delivered to the stroma? As we discussed previously, carbon dioxide diffuses through the stomata on the bottom of the leaf into the stroma of the into the leaf, into the mesophyll, and into the chloroplast to participate in Calvin-Benson cycle. Let's take a look at the process of carbon fixation. So it all starts, and it's weird to say it all starts since it is a cycle, but it starts here when the enzyme called Rubisco, ribulose 1,5-biphosphate carboxylase oxygenase, a mouthful, facilitates the reaction between carbon dioxide and ribulose 1,5-biphosphate. So 3 molecules of ribulose 1,5-biphosphate contain 15 carbons. Each molecule contains 5. 3 molecules contain 15. 3 molecules of CO2 contain 3 carbons. When 3 and 15 are combined, they form eventually 6 3-carbon molecules of 3-phosphoglycerate. So still we keep 18 carbons together. Now, the molecule of ATP is used to convert phosphoglycerate into 1,3-biphosphoglycerate, and then 1,3-biphosphoglycerate is converted to G3P. The most important step, probably, although it's hard to say which one is most important, but the first step here is called carbon fixation, because essentially during this step, inorganic carbon is fixed from its inorganic form into organic molecules of 3-phosphoglycerate. Strictly speaking, one turn of the cycle fixes one molecule of CO2. One molecule of CO2, one carbon, reacts with one molecule of RuBP, 5 carbons, forming 2 molecules of 3-phosphoglycerate, 6 carbons. So carbon balance is kept. Now, second step is the reduction step. During the reduction step, 6 molecules of ATP and 6 molecules of NADPH are used to reduce 3-phosphoglycerate to G3P. ATP is first used to phosphorylate 3-phosphoglycerate to 1,3-biphosphoglycerate, which is then reduced by the electrons coming from NADPH to the molecule of G3P. The molecule of G3P must be familiar to you because it's an intermediate during the process of glycolysis. In the meantime, ADP that is produced when ATP loses its phosphate and NADP plus that is produced when NADPH lose electrons, they return to light-dependent reactions to be reused to produce new batch of ATP and NADPH. Now, since it is the cycle, we need to figure out how to come back from that step to here. So this phase of the cycle is called regeneration. During only one of all the G3P molecules, in this case, in this cycle, 6 G3P molecules is taken away to produce glucose or other organics. And since we mentioned that one turn of the Calvin cycle fixes one CO2 here, each turn of the cycle fixes one CO2, you need 3 turns of the Calvin-Benson cycle to produce one molecule of G3P. Now, 5 molecules of G3P remain in the cycle and used to regenerate ribulose biphosphate. For this regeneration, so this is 5 molecules of G3P, this is the regeneration step. For this regeneration, 3 more ATP molecules are used in these reactions. Which brings us to the concluding idea. The energy flow in the biological systems. What we learned about is that in photosynthetic organisms, light energy is absorbed to build carbohydrates from inorganic carbon, and oxygen is produced. This oxygen and the produced sugars will be used in mitochondria of heterotrophs and autotrophs alike to generate ATP in the process of cellular respiration and produce CO2 as a byproduct, which will come back here and will be reutilized again. The similarity of these two processes is staggering. Both of them use the electron transport chain to capture the energy. Chloroplasts use electron transport chain to capture the light energy, while mitochondria use the electron transport chain to capture the chemical energy stored in the sugar molecules.